Good morning. So talk a little bit about soil health and just look at this picture here. If you look at the one on your left, you see lots of signs of soil health. You see good tilth in the soils. We want that chocolate cake or coffee ground structure that Clint talked about. You see earthworms. There's no way better to build soils than with a good sod grass crop, but that doesn't always pertain to row crop farmers. But if you look at the picture on the right of corn roots, corn plant is a good indicator of soil health. And what we're looking at is all the soil clinging to the roots. Why does that happen? It's because a healthy plant will put out root exudates, and the microbes feed on those root exudates and colonize on the roots. And when that happens, the soil clings to the roots and you can shake it and it doesn't want to fall off. But that only happens in the aerobic zone, or the soil that has oxygen like Clint was talking about. So once you get below the aerobic zone, you end up with bare white roots. So the corn plant can tell you a lot about what's going on in the soil. So Clint talked about this. What does the destination look like? It's the combination of the physical, the chemical, and the biological coming together. The definition from Wikipedia, it's a state of composite well-being in terms of biological, chemical, and physical properties. Going to move on here. Benefits of a biological active soil. We got nutrient cycling, and that's the big one. We can get nitrogen fixation, convert organic nitrogen into plant available forms, solubilize phosphorus and other nutrients, slow release nutrients. Oh, there we go. As microbes consume nutrients, they're contained in the body, and they give it up for another microbe or the plant to use. So in a sense, your fertility or nutrients become time released through the work of microbes. And this allows us to use lower rates of purchased fertilizer. And ultimately, we want to build organic matter. Clint hit this pretty well too. If we have a healthy biological soil, we're going to have tilth and aggregation. That allows more water infiltration, root penetration. We can hold on to more water and nutrients. Some more benefits: can infect insects and nematodes to keep them in check. We compete with fungal and bacterial pathogens, and we do that by having beneficials there to outcompete them for the food sources. So they take over. So how do we get more biology in our soil? Well, we can inoculate. For one thing, we can inoculate soils. We can inoculate seeds with seed treatments. We can inoculate plants by putting biological products in the root zone so they can colonize those roots and have the beneficial organisms there helping to cycle those nutrients. And then we can inoculate our crop residues so they break down quicker and again cycle nutrients faster and help us to build carbon in the soil. This is a chart put together by Dr. Robert Kremer at the University of Missouri, and it shows just the diversity of microbes. And we want diversity because each microbe brings something different to the table. You see here the different functional groups, promoting plant growth, disease suppression, weed and insect suppression, nitrogen fixation, soil structure, carbon cycling. So there's all these different functions that microbes can do, but we need a diversity of microbes. I kind of compare it to a potluck dinner at church. You know, everyone brings something different to the table, and it turns into a pretty pleasant eating experience. If we have a diversity of microbes in our soil, it turns into a pleasant crop growing experience. So we want that microbial diversity. So some of the microbes that we work with in our products would include bacteria and fungi. Some of the common bacteria are Bacillus, Pseudomonas, Streptomyces. Some of the fungal organisms are Trichoderma, Penicillium, Aspergillus, Mycorrhiza. And there's literally hundreds of others, but these are some of the more common ones. So this is kind of a soil or a life pyramid, and human life is at the top of the pyramid. It is the most complex life in all of creation. But if you look at the bottom, it starts with the more simple organisms like bacteria and actinomycetes. You know, a lot of people talk about fungal organisms, and they're certainly important, and they want fungal-dominated soils, but we really have to have that base of bacteria and actinomycetes in the soil as well. So what happens when microbes run out of food? Cellular metabolism slows down, they can revert into a dormant state, and some may even perish. And that's why we need to re-inoculate from time to time, because all these conditions happen. You know, some people think if you put the organisms out there once, you should be good. You know, you've re-inoculated the soil, but environmental conditions change. You know, soils become too dry, they become too wet. We run out of carbon in the soil to feed the microbes. So that's why we need to continually feed microbes and to re-inoculate. And one of the best ways to feed the microbes is through root exudates. And that only happens when we have a crop that's well fed. If the crop doesn't have adequate nutrition, it doesn't have enough energy to put out root exudates and feed the microbes. So how do we reduce the risk of not having the right microbes? And that's why we provide diverse biological products. Like our bioenergy is our flagship liquid biological product. We've done DNA testing on it, and I think there's over 300 species of bacteria in it and over 100 fungal species in it. And then the Endo 25 that we use for a seed treatment, it has many the same organisms, but it also has mycorrhizal fungi. And you've probably all heard of mycorrhizal fungi. They act like an extension of the root, helping to solubilize phosphorus and other nutrients in the soil. So again, we want that diversity of microbes, so we're covering all those functional groups, so our soil functions at maximum capacity. So over time, microbes produce aggregate soil structure. And that's because they secrete a polysaccharide, which is kind of like a biological glue that takes individual soil particles and glues them together. And that's what gives us that coffee grounds or chocolate cake crumb structure that we want to see. And once we have that, it allows air to enter in the soil and roots to freely penetrate. And we want those microbes to colonize the root rhizosphere, or that's the soil immediately surrounding the root. You know, an acre of soil plow depth has 2 million pounds of soil, but your roots actually only touch a very small percentage of that. So the real activity happens in that root rhizosphere, that area immediately surrounding the root. So if we can change that environment, we can dramatically change the impact that the crop has. We want microbes to raise the level of nutrient availability and plant uptake. And if we have a healthy, biologically alive soil, it can definitely decrease the amount of nutrients that we need to purchase to grow a crop. So we want to time our fertilizer inputs and avoid over-applying at certain times and not overload the system. This is a picture of the agri-energy farm there, and you see a line in the soil where it's darker. It's a fence row, the agri-energy farm versus neighboring farms. And that picture was taken in the late 90s, I believe. And those farms, the neighboring farms, have similar tillage practices, similar crop rotations. Why is the soil darker on the agri-energy farm? Well, it's got more organic matter. Why does it have more organic matter? It's because we've been working with biology, using microbes to speed up residue decomposition, applying microbes with the planter. So essentially, we're building our soil biological diversity. We have a healthier biological soil that's creating more carbon, more organic matter. And that shows up in the picture very plainly. Oh, now we got the line there, but that's the only difference is, the cropping practices. This is from the state of Texas in the Panhandle cactus. We have a dealer down there, and this is the change he made in a soil in 1 year's time. We've got one pivot that was treated and one that wasn't, and this literally just several feet apart from each other. I was actually on that farm about 3 years ago, and that's the impact he was able to make in one growing season by using some biological inputs. You can just see a huge difference in the soil tilth there, the color of the soil, the texture. It's very dramatic. This is a picture of one of our dealers, Ray Rutger, in southern Indiana down at Boonville on his own farm there growing sweet corn. Ray passed away here several years ago, but this is the 5th year he grew sweet corn in the same spot, and the only thing he put on was SP1 and a little bit of molasses. So this is year 5 with no other inputs than SP1, which now we would use our bioenergy. But he was using like 30 gallons per acre and a little bit of molasses with it. So it kind of illustrates what is possible just with microbes. So conditions that influence the growth of soil organisms. The amount and type of nutrients. And carbon is a big one. Carbon can be sugars, it can be humic acids, it could be fish, amino acids, and root exudates. And that's the big one. Available moisture. Microbes, for the most part, or at least the beneficial ones, like the same amount of moisture in the soil that the crop does, 50 to 80% of field capacity. If it's more drier than that, then they don't function well. If our soils are waterlogged, that tends to suppress their activity, especially the beneficial ones. Some of your pathogens can flourish in those saturated conditions, but not the beneficial ones. Degree of variation. We do need oxygen in the soil, at least 2 parts per million. And that comes back to that air and water management. We need a soil that can breathe, so we want air in it. And then temperature, 70 to 90 degrees. If it gets warmer than that, It's going to limit the activity of the microbes. And if it gets below 70, it's going to slow down some. When soil temperatures get below 50, it slows down even more. You know, that's why they say wait till it's below 50 degrees to put anhydrous on. They don't want the microbes to convert that ammonia into nitrate. We do have some organisms that function in the 30-degree soil temperatures, but these are the ideal parameters. And then a pH of 6 to 8. But I think that's important to remember when you're putting on biological products. What kind of environment are you putting them in? You know, is there enough moisture there to sustain them? You know, if you're spraying a biological product on the surface and it's 90 degrees and sunny and windy, you know, what's the chances of survival? So if we can get microbes in the soil, their chance of survival is much better. It's just important to keep this in mind when you're applying products. You know, what kind of chance of success am I giving these organisms to survive and thrive?
A real quick question while you're there, if you don't mind. On that survival rate, you know, we obviously have had a drought for a couple of years, and so we go to spray, you know, are we better to wait until toward evening, or is overnight a little bit of dew enough to sustain that, or is it just, you know what, let's just hold off and try something later down the road?
Yeah, I would say if you're spraying a biological and it's only going to be surface applied, anything you can do to enhance the moisture, spraying in the evening and hope for a dew overnight, or if you can time it in front of a rain, it's going to greatly enhance your success. Then root extensiveness. Again, we want to proliferate microbes on that root rhizosphere. So the more roots we grow, the more chance we have to feed microbes and bloom their populations.